All right, everybody, I think we can start. So thank you all for coming. This is my second time here in Austin, so I love the festival. I love everything that happens here. So it's my big pleasure to be here, and I assume that you are here because you want to understand how to reconstruct the human brain inside the lab, and that's exactly what I'm going to tell you. So I'm going to tell you how to do that, and I'm going to tell first why we do that, and what are the reasons that we do, and how can we apply that to different types of technology. So let's start with with why. The reason that I do this is because I have a son with severe autism. So my son doesn't talk. He cannot communicate. He's 18 years old now, and he has like severe types of seizures. So he requires attention 101, 24/7. And that's hard on him, that's hard on the family, and I'm not alone. There's several families with kids with severe, or we now call profound autism, and this is a big deal. And similar to autism, there's lots of neurological conditions with no good treatments, no cures. And while we see lots of advancements in medicine, for example, in cancer biology, there's subtypes of cancer where we can have like a cure or a treatment. Nobody's dying anymore from heart problems, but the brain continues to be like a major health issue. And the reason why is because we are lacking like a good human model. We don't study the human brain on human brains. We study the human brain using animal models. Mice is the favorite animal model for biomedical research, but the mice evolve in a very different way than the human brain. So the whole idea was to move from what is causing these conditions, and we know that there is a strong genetic factor, to how that mutations in your genome can actually cause all these behaviors that leads to all these neurological conditions. So to do that, we need like a good model, and as I pointed out, animal models are not doing the trick, so human model would be the best thing to do. But there is a problem, and that's the reason why scientists do not study the human brain. First of all, we don't know how the human brain is formed because it's inside the uterus. And there is no invasive technology that can actually look at the formation of the brain. We don't know when the first neuron fire. We don't know when the first synapses are formed. We don't know how the network shapes behavior. So everything that we feel, we don't know how it happens. We don't know how we retrieve memories. So these are all intrinsic into the brain, very fundamental questions, and we have no idea because it happens inside the uterus. And even in the adult brain, you can say, well, let's study the adult brain. The adult brain is inside your skull. Again, we have no non-invasive technologies that can get into the brain, look at the specific synapses, and trace all the circuitries in there. So that's why we use stem cell to recreate the human brain. So this was a first proof of concept created by a Japanese researcher called Yoshiki Sasai in 2008. That's when I was starting my lab as well. And what he shows is that you can take stem cells, and these are the kind of stem cells that can make different tissues. We call them pluripotent stem cells because they can make all tissues in the body. And they can guide with specific factors these cells to form brain cells. And this was the first proof of concept, very difficult to reproduce. It took a while for other people in other labs to start getting their hands on. So, but before I continue, let me just make sure that we are all on the same page, because you're going to hear in the media tomorrow that someone is creating like mini brains in a dish. And it's not exactly that. It's not a miniaturized version of the human brain inside the lab, but it rather is a piece of the tissue. And there are limitations that are intrinsic to this technology. For example, these structures that we call a brain organoid, they are not vascularized. And that's the reason why they don't grow bigger. They can reach 0.5 centimeter, but they cannot pass to that because the nutrients are not getting inside the tissue. So the neurons that we have are quite immature. We are just learning how to induce maturation on those neurons. That is also a big limitation. And as we, scientists, are increasing this technology, there is going to be ethical and moral questions about that. And I'm sure after the talk, you're going to understand what I'm saying here. So these are limitations, but let's see what we can do with the technology that we have right now. So In 2010, we start optimizing the protocol developed by Yoshiki Sasai to create something easy that many labs in the world could do. So we simplified the protocol in a way that there is a couple of steps, and these steps are getting cells that are single cells, dissociated pluripotent stem cells, and neuralize them by adding some factors. So there are two factors that we add. One factor blocks the differentiation of all the tissues but the brain, and the other factor stimulates the differentiation of the brain cells. So when we combine those two factors, the cells neuralize and form what we call a neurosphere. So that contains lots of neuroprogenitor cells. These are the cells that will give rise to all different parts of the brain. So after that, we stimulate them to grow, and as I said, they grow to a certain point, and then they stop growing because the tissue is not vascularized. And after that, we remove these growth factors, and we let those spheres mature. And they can mature for several years. The record in the lab is about 3 years. We just couldn't continue because someone dropped the plate. It happens in science, right? The human error. But in theory, we can keep these structures for life. I mean, they should last the same as the human brain. They should last like an entire life, as long as you keep taking care of them. It's like little bonsais. You need to go there every day, check it out, clean it up, and they will continue to thrive and mature. But nonetheless, I mean, if you haven't seen it, this is Isabel, a postdoc in the lab, and she's handling like a plate with all these brain organoids in there, and each one of these white dots is one of them. These are very early stages. This is how, after like a couple of months, so they grow, you can actually see it. It's thick at naked eye. You can put under the microscope. So they go from single cells, and they can reach up to 5 million cells in there. So in each one of these, you have like roughly 5 million cells. 2.5 million neurons are in there, equivalent of a bee brain. What I haven't mentioned to you is that this technology works with any live person, meaning that I can do this using cells from you. And usually I get cells from the skin or blood, and then I reprogram the cells, and then I make these mini brains. So these are going to be avatars of your brain during development. So in a way, I'm going back in time to replay your neurodevelopment in the lab. So if I can do that with kids with autism, for example, I can compare with neurotypicals and see what goes wrong, and perhaps I could help to fix or could help to treat. So that's the whole concept, right? I mean, the way the cells are formed, these are the biggest surprise that we had in science. This is a two-dimensional representation of these organoids. On the top will be the center of the sphere. And I hope you can appreciate that some of these progenitor cells are migrating from inside out. And they do that by using cables. There are some cables in there. So these are specialized cells that helps on the migration of these progenitor cells from inside out. And as they get to the periphery, they start to differentiate and forming network and connections with the neighbor cells. If you slice one of these spheres, you're going to see a structure like that. You have a ventricular zone inside where most of the progenitor cells are there, and they migrate out forming these layers. So the cortex, the human cortex that controls your behaviors, that controls your feelings, are composed of 6 layers. So this is just the beginning of how the human brain is formed. And they do exactly as we see in nature, from inside out. And as you keep maturing them, they will generate a diversity of cell types, and these are the same cell types that we see in the human brain. If you're not familiar with this graphic here, each one of these dots is a cell. We just take one of these spheres and we explode them, and we look for the markers to tell us which cell types are there. So the pool of progenitor cells are there in pink. They're producing mostly glutamatergic neurons. These are your excitation. That's how the human brain is formed. The early periods of gestation, your brain is full of excitation. And this is at 4 months, where they make most of these excitatory neurons. On top, you see that there is a discrete population of what we call inhibitory neurons. This is what's going to balance your brains. So the right combination of excitation and inhibition makes your brain. So if you have unbalance of that, you might have seizures, or you might be like in the end of the spectrum for autism. So that combination of excitation and inhibition is really important to balance your brain. At this stage, we also have in red the glia cells. The glia cells come from Latin glue. These are the cells that intercalate with neurons and help the neurons to form the connections. So the synapses do not form without the glia cells in there to actually shape it. So at 4 months, everything is happening. And at 4 months, that's exactly at 4 months of gestation, of human gestation. So it follows the neurodevelopment. But the big things for us is, can they actually form real networks? And that was a major bottleneck that we could only solve in 2019, where we start plating these organoids on top of multi-electrode arrays. And I'll break that down for you. So the organoids, or the spheres, are placed on top of electrodes that are printed in the bottom of a dish. So all the time when there is electrical activity passing by, the electrodes will capture the activity and generate an activity map, so you can see it flashing. So these are the neurons firing. And you can do that recording over time. So this is creating this raster plot. And scientists, by looking at these raster plots, we can see each channel when they fire. And we can see when they fire together as an indication that these networks are being formed. And to our biggest surprise, what happens is in 2 months, we don't see any organized network. So there is only random spikes being formed. By 4 months, we start to see some level of organization. We start to see the emergence of neural oscillations, suggesting that different neurons are talking to each other in different parts of the organoid. By 6 months, it is all there. It's all interconnected, but it's still very stereotyped. By 8 months, they become highly complex, and the complexity just increases from 9 months on. And I'll tell you exactly how they look like. So we run into a problem at this stage because I started by saying how hard it is to study the human brain. And when we showed that our organoids could actually make these neural oscillations, these patterns of electrical activity, the entire community started pointing out that we don't know if this is how the brain works, because nobody measured electrical activity in uterus. So we have to rely on premature babies' brains, where we place the electrodes on top of their heads, and then we could record from them. And so they generate what we call an electroencephalogram, which shows the waves of their brains while they are developing. And different from an adult EEG, the neonate EEG has a very typical or very stereotyped behavior. It shows peaks of activity and lots of silence time. And as you grow older, as you receive input from the outside, the periods of quiescent, where the brain is silent, goes away, and your brain becomes highly interconnected. So we use that as to create like a machine learning process where we map, where the machine was learning based on the EEG, how to map the age of the subjects. And we did that with premature babies starting at 25 weeks up to post-gestation, or 35 or 40 weeks, 42 weeks, about 9 months. Once we map all the EEG, we start feeding the machine with the electrical activity coming from these structures made in the lab. And the result was amazing. The machine gets confused. It cannot distinguish between the brain, the real brain, or the brain made in the lab. So this is how they look like. The predicted age is in one axis, the actual age is the other one. The perfect correlation is the dotted line. The human brain is in black, and the organoid is in blue. Each one of these blue dots is the recording from one organoid. And although they seem more variable, they all follow the same trajectory, meaning that they are maturing, they are forming these networks in the same way as the human brain. So you can imagine, I mean, if these crazy scientists build like a piece of a human brain that now behaves like the human brain, what happens, you become news, right? And that's what happens. You become like cover of the New York Times. You create like the human brain in a dish, right? But that comes with a cost. And that's the cost of the ethical and moral issues that accompany this kind of a research. So lots of people start raising red flags. Is this ethical to recreate your brain in a dish? And we can move on to that discussion later on, but let me start saying, why do we use this for? And I guess you guys remember in 2015, 2016, there is an outbreak of Zika virus in Brazil. By the time we have the babies with the microcephalic brains, we didn't know exactly what was the causal agent for that. There is this correlation with the Zika virus, but there is another possible interpretation, could be like a pesticide that they were using in the northeast of Brazil. So the only way, the only tool that could actually prove causation was the human brain organoid. When we place the human brain organoid in contact with the Zika virus, it actually kills the progenitor cells and the cortex reduced. That's exactly what happens in the babies of Brazil. So that was one application that we could even use that to screen for antiviruses that could be repurposed to treat those kids. Fortunately, it was easier to control the mosquito who was transmitting the virus, but in case of a new outbreak, and by the way, this type of mosquitoes are getting to North America, we have drugs that now can prevent the vertical transmission from mom to the fetus. So if there is a pregnant woman now positive for Zika virus, she might take one of these FDA-approved medications and protect the fetus. So this is one possibility. So the Zika virus actually travels from the mom blood and gets into the uterus, and that's when it causes the problem, right? But there's lots of chemicals that do the same thing. They travel from mom to the uterus and affects how the brains form. And believe it or not, we don't have good models to do this kind of a toxicology. So we thought about, okay, what if we start testing the environmental factors that we have here and see how much they can disrupt the formation of the cortical networks and create what we call like a brain-safe label. So this is what we are proposing for the toxicology from now on. Instead of just testing the toxicology in mouse models, we should be using a human model as well. And this human model will tell if the chemical that is getting into pregnant woman is safe or not. And just to illustrate some of the questions that we have, I believe you heard about acetaminophen, Tylenol, given to pregnant women, highly associated with high levels of autism. The same thing with PFOS. These are the long-lived molecules that are hydrophobic. You find it on your pen, your Teflon plane. You find it on their carpet. That's exactly where we put the babies on. So we all have these molecules in our bodies. How bad is that? How much is tolerated by the human fetus? So we can answer all these questions with this human model. So that's why we are big fans of that. But let me go back to autism spectrum disorders, ASD. Can I do something for these kids? Can we actually use this technology to find medications? And the first type of autism, well, the first challenge that we have was to get the families back into the lab. Because if you have kids with profound autism or very severe syndromes related to autism, they cannot move. It's hard. It's hard for the families, it's hard for the parents. So we create a social network where we deliver what we call the Tooth Fairy Kit Collection, which is a small tube with media where we ask for the milk tooth from the families, and they would put in there and send it to the lab. So the kids, they don't have to come to the lab for us to isolate a skin sample or a blood. We just get the cells from the dental pulp, and that was enough for us to get started. This program was so successful that it started like in 2010, 2011, and up to today, even though we close it, we still get teeth or tooth from families. And there was a learning curve how to do that, because in the beginning, the families were washing it so well and removing all the cells that we need. So we have to tell them, don't wash it, just put it on the tube and send it back. But hopefully, I mean, yeah, finally, I mean, it was successful, and we get enough cells from kids with autism, so we start looking for differences. And this is one of the major differences that we see. It is coming from a syndrome under the umbrella of autism called Rett syndrome. And what you are seeing here is a network of neurons. The process of those neurons is in green, and the synapses, the connections between neurons, is in red. And I hope you can appreciate that the Rett syndrome neurons, about the same time, they're not making enough synapses, enough connections to actually compare to neurotypicals or control neurons. So by showing that, we show that, yeah, we are recapitulating something that is unusual on their brains, and we can quantify that. But not only quantify, then we ask a second question, is this revertible? And this was, at the time, was going against one of the dogmas of neuroscience, saying that if you pass the critical period, your brain no longer can be reverted back to a normal or neurotypical state. But what we show is very interesting. We add a molecule called IGF-1, insulin growth factor 1. Neurons love it. They love it so much that they start making connections. And we were able to bump it up, the amount of synapses that was formed in these networks. So long story short, after 10 years, this becomes like the first treatment for Rett syndrome. So the problem that I have now is the clinical trials are taking too long. And this is because we fail and we have to do it again and the molecule doesn't work, we have to start again. But if you have a kid with autism like me, you want to see it, the solution faster. So I cannot wait another 10 years. So we are using this technology to kind of speed up the whole process of clinical trials, trying to find proof of principle as soon as possible and then move into testing directly into the subject. And this is just like some of the syndromic forms of autism that we are testing. So we create these brain organoids and we see what's wrong, and then we try to ameliorate the phenotype or ameliorate the condition using pharmacological treatment, gene therapy, or any other tools. We are agnostic of the tools. Whatever works, we're going to test, and then we can move into clinical trial faster. And on the gene therapy, if you know the gene that is mutated, this is the part that is moving super fast. Once you know the gene, and you can easily get done in a genetic lab that just sequence your entire DNA or the DNA from your family and point out what is the mutated gene. If we know what is the mutated gene, we can create what we call a viral vector, which comes from a flu virus that doesn't do harm to you, but we make it, the virus carry the correct version of the gene back to the brain. So that's what we call a gene therapy. And we've been testing this for different types of genes associated with autism. And we're having very successful cases. So this is for Pete Hopkins. We can show that their brains were not fully formed, but then after the gene therapy, you can start seeing the neurons reconnecting, and even the neurons that were not there start to emerging. Malformations in the brain that we never expected to see coming back are coming back. So it's challenging what we know as a neuroscience, because we always thought that, well, I mean, if the cells never emerge, they will never come. But no, after gene therapy, if you just place the gene back, it reverted back to a kind of a more neurotypical state. Because we're so successful, we had like a donation recently from a family from Orange County for us to start a gene therapy program here at the university, where we can go from the discovery towards the clinical trials in a single shot. And we discussed with the FDA, and as long as you have the proof of principle, yes, we are clean to go. So this should speed it up now, all the processes to get into the clinical trial, so we don't have to wait another 10 years like we did in the first time. All right, so remember when I mentioned that when we make these structures in the brain, they follow neurodevelopment. So if they are a 4-month-old organoid, they are equivalent of a 4-month-old organoid, they're equivalent of a 4-month gestation. So the model is so good that it follows exactly the neurodevelopment. But sometimes we can't wait. Sometimes we want to know what's going to happen to that kid when it becomes 18, when it becomes 25, when it becomes 80 years old. And I've always been looking for ways to age the brain cells or these brain structures in different ways. And one thing that we never did was to stimulate them. So once we grow these organoids, we put them in an incubator and they stay there. No sensory information. But after 6 months, this is not what happens to your brain. So you kind of you're born and open your eyes and start receiving all the sensory information from the environment. So we start thinking about what if we start plugging things in the brain? And I got the idea from my kid, actually, because Ivan, my kid, what he does, he starts combining pieces of stuff in the house to create what we call sculptures. And he's getting good at that, and now these are 2 pieces. Now he finds that if he uses like a rubber band, he can plug more things together. And I found it so beautiful, so we just keep taking pictures. And I thought, well, but maybe he has a point. Maybe we need to plug sensory information in the organoids, and that's how we can mature it. And so I'm going to show you like some of the stuff that we're doing for the eye. So the same way that we can create the brain, we can create the eye. And this is like a 3-way organoid where you have a retina organoid, we have a thalamic organoid, because that's where the projections from the eye pass through the thalamus before it reaches your cortex, and you have the cortex. And you can visualize the different pieces because we place fluorescent reporters so we can see it. So these structures are now maturing in the lab, and you can see a very nice photoreceptor in there, and all the axonal projections passing through the thalamus and reaching the cortex. So once we have this kind of a structure, we can start giving them visual cues. We can start stimulating them. And so that's what we do in the lab. We have this kind of a tools that keeps blinking different frequencies and intensities in these structures while we record from the cortex. So can they see something? Can those structures made in the lab see something? So we did that in 2 different ways. The first way was with my colleagues, Dugil and Anna Devour. What we did was we removed the visual cortex from an animal, and we replaced with this structure from humans. And once we remove the visual cortex from the animal, it cannot see it, right? You present visual stimulation, and the animal cannot see it. But now, once I replace with the human tissue, start seeing again. So the animal is seeing the world using human neurons to recreate their network. So that's a proof of principle that the visual system can be recreated. But what about in a dish? Can those things see in a dish? So what we did was, again, the same thing. I mean, we stimulate from the bottom while we record from the top. And because we are giving like different frequencies and intensity, we keep looking for when these mini brains or these organoids react to the right stimulus. And it's so funny because there are certain things that they don't react at all. We keep like stimulating, they don't do anything, but there are certain frequencies that they react. Some people in the lab like to call that a reward. I don't like that because I don't know if they're liking or disliking it. I just call it reacting. So they are reacting. And as they react, we see something very interesting happening. We see that some of the synapses are becoming stronger and forming like the right circuitries, and other synapses are being eliminated. So this is the basis of neurosciences. This is the basis of learning and memory. That's what we call LTP and LTD, long-term depreciation, long-term potentiation. And these structures, man-made structures, already show that. All right, so if we can give them like sensory information, can we use that to become part of artificial intelligence? So this is another thing that we could perhaps use these networks, and we call them organoid intelligence. So we are using how the human brain computes to create a different form, a different type of AI. And so there are many reasons to do that, but mainly because the human brain is primed to learn, and we learn in a different way. So everything that we know about learning happens after we are born. So that's how we study learning in humans. But humans are born with tons of genetic information encoded in our DNA on how to intuitively learn things. And we want to learn how the human brain learns. So we are preparing experiments to do that. So this is how AI learns, right? So it requires training, tons of energy to train things. And not all the times it gets right. So that's the hallucinations that we see in all these tools now. But the human brain doesn't do that. So this is like, if you teach a kid, I mean, who is your dad, or what is your, I mean, what's a toy, what's a ball, it gets it. You don't need like lots of training, just once, and it gets it. And so we think that perhaps if you use the organoid as a computational power, a computational tool, we can solve some issues, for example, energy problems, because the brain doesn't take too much energy. It's actually quite efficient. Data storage. Look how many memories we have collectively in this room. I mean, we can solve memory problems. Continuous learning, and we can improve and even create a more humanized AI if we do that. And I have colleagues here that are doing something even crazier. We are stimulating the quantum computing on these organoids. So if you want to learn more about that, come talk to me later on, because we think that we can revolutionize the way AI currently works. All right, so but let's go back to reality. What we can do now, and what you are seeing here, it is the first cyborg. So this is like a robot that is learning how to walk using the networks created by one of these organoids that now live inside the incubator. So the electrodes from the multi-array is collecting the information and is feeding the robot and teaching them how to walk around. And we are now putting sensors on this robot. For example, these two eyes, these are infrared, that are creating like a feedback loop. So it tells the robot when it's approaching an obstacle and has the opportunity to turn left or right. So by teaching these organoids how to move this robot, we are teaching them how to avoid obstacles. So now we can place this robot in different mazes and see how they react. And this is by learning just once. And they can do that quite efficiently. So why can we apply this technology in medical sciences? In autism, for example, and this is true for Alzheimer's as well, we have what we call cognitive resistant or cognitive rigidity. That's when people have hard time to learn or deny things. So in autism, we see on the most severe side of the spectrum, we often see some problems on cognitive rigidity, difficulty to learn. So we are reproducing these experiments now in the two settings with neurotypicals and people with cognitive rigidity to see what is the problem, why they cannot learn as fast as a normal neurotypical brain. And once we find out, we can start the treatment. And one of the treatments that we are very intrigued is on the use of psychedelics. And we are testing some psychedelic molecules such as DMT or psilocybin to open up the learning opportunity of the human brain to treat people with cognitive rigidity. So this is the data from DMT. Don't need to pay attention to these graphics, but just to say that in the presence of these psychedelic molecules, the brain starts to open up and start learning much faster. And that can be applied to kids with autism, for example. I'm not suggesting to give psychedelics to everybody, right? There are a different class of psychedelics now that can be created without the hallucinogenic effect, although that might create other problems, but it remains to be seen. But there are lots of LSD molecules, for example, that doesn't keep you high, but allows for your brain to have this possibility of learning again. And these are the type of therapeutics that we want to see moving forward. So there's another part of the lab that wants to understand why the human brain is so unique. And I have this question since I was a kid, because I was always seeing like the other animals and how they behave. And we humans are so different. I mean, we move around the world, we change the world, and this has to be what makes our brain so susceptible to some of these conditions. You don't see autism in animals. You don't see Alzheimer's in animals. Even chimpanzees that has been observed in the wild or in zoos for a long time, they don't show signs of Alzheimer's or dementia. These are things that are human specific. And to get into the bottom of why, what makes us human, we decided to compare ourselves to our close relatives, the Neanderthals. And different from the Neanderthals, who in a very large period of time that they were here on Earth, we, in a very short period of time, moved from stone tools to all this technology that we become used to. So if they have about the same size of a brain of us, if they're so similar to us, why have not the Neanderthals create all these type of technologies? And they have all the time to do it. So I think you can see where I'm going. Oh, this guy's going to make brain organoids from the Neanderthals. The problem is there is no Neanderthal cells. We can get their genome, but we cannot get their cells. The cells do not survive all that time. But we have a genetic hypothesis, which is we, modern humans, we accumulate mutations in our DNA that make our brain so special. And if we can find those mutations, we can probably understand how evolution creates a brain that is so unique like ours. And so we scanned the human genome looking for regions that are different between us and the Neanderthals, and we end up with 61 genes. The human genome contains 25,000 genes. There are 61 genes that are different between us and the Neanderthals. Among those, there are genes that are related to bone mass and muscles, and of course that matches with the fossil records. But there is a couple of genes that are quite interesting because it affects neurodevelopment. And one of them is called NOVA1, and it's a master regulator of neurodevelopment. It happens very early when your brain is being formed and affects how you orchestrate your balance of excitation and inhibition, for example. So since we cannot have the Neanderthal brain, we can edit the human genome to carry the Neanderthalized version of the NOVA1 gene and see how those organoids will behave. So in a way, it's resurrecting an ancestral, resurrecting an extinct genetic variant that nobody has here, but we can place that inside one of these organoids, and we can look at them, and they look different. They actually have this popcorn shape, suggesting that the migration of the cells happens in a different way. But more interesting, when we analyze the electrical activity of these networks, we see that the Neanderthalized brain organoid matures way faster. So it matures 10 times faster than us, than normal humans. But there is a price for that maturation. It is a lower level of complexity. So you increase the maturation, but decreases complexity. And in the beginning, I thought that this was counterintuitive. But now looking at baby chimpanzees, they outsmart human babies because they have this faster maturation. So this mutation in this NOVA1 gene slows down human maturation to a point that generates more complexity. And we think this is the basis of modern human conditions, is the fact that we slow down human development. All right, so if I can use genome editing technology to look into our past, to resurrect ancestral mutations, can I use to look into our future? Can I recreate a human brain that never existed? Can I create brains that are resistant to Alzheimer's, that are resistant to dementia, right? So these are the things that we are doing in the lab. And one of the projects is also to create a human brain that is resistant to cosmic radiation. And we do that by adding a gene called DoSoup that comes from a tardigrade inside them so they can become resistant to cosmic radiation. And this is the kind of a project that took me to collaborate with NASA and grow the brain organoids at the International Space Station. So we have like several missions now. We call them payloads. And it works like that. So first of all, why NASA is interested in that? Well, because if you have long-term astronauts, when they come back to Earth, they are all messed up. I mean, all the different tissues have a very systemic problem because they seem to age faster in space. And this is a big problem if you want to go to Mars, for example, which is a goal of NASA, is to go to Mars, right? How the human body will cope with that, how the human body will adapt to microgravity or to this space environment. And this is the data from NASA. This is just on human cognition. So it's a project called the NASA Twins Project, where you have 2 astronauts. One stayed at the space station for about a year. The other one was the ground control. And after 1 year, they compare the different tissues, but this is measuring human cognition, learning, capability of reaction, memory. And this graphic shows pre-flight, during flight, and 6 months post-flight. And basically tells that even after 6 months post-flight, the cognition of the astronaut is really lower compared to the normal controls or even his brother, suggesting that something happens in the brain that affects the way cognition normally behaves here on Earth. And is dramatic. There's something really bad happening on the brains of astronauts. And we need to figure this out if we want to have like interplanetary colonization. So the experiment that we do is the following. This is just a summary of one of the experiments. We grow these brain organoids in San Diego. We use like an autonomous box. I'll show you in a minute. And then we go to Florida to the Kennedy Space Center, where we assemble everything. And then we place that into a Falcon 9, the SpaceX rocket. And we ship it to the International Space Station. They stay there for about 30 days. And after that, we put back on the Dragon capsule and splash down the ocean, go with a little boat, collect it, and bring it back to the lab. So this is the type of payload. We have all the organoids in those tubes in there. It's the size of a shoebox, contains the battery, all the microfluidic chambers to keep these organoids alive. So it really doesn't require too much for an astronaut. The only thing that the astronaut has to do is really to take our box and plug inside one of these cabinets at the International Space Station. As soon as it's plugged in, I can actually see it from my laptop. We have the last payload coming back in last December, and I was on the beach just checking out. But yeah, come back, good, nice. So this was all very exciting. But what did we learn from these missions? What happens to the brain cells when they come back from space? And this was a big question because it's affecting astronauts' brains. So we learned that, yes, we see this accelerated aging after 30 days. And it's not linear. It doesn't happen in the same way that happens on Earth. There are some molecular clocks that are hitting 10 years, and there are some molecular clocks that are hitting 80 years. So there's whole messed up at the molecular level, and we want to understand why. Because it's a new phenomenon. We call it space-induced neurosenescence, or SINS. So we can distinguish from the normal aging that happens here on Earth. It is something that happens in a very compressed time. In 30 days, you are showing signs of a very old brain. And we see that at the molecular level, at gene expression level, so we are starting to learn how this phenomenon actually started. And I'll show you one example of how can we use that, not only to help the astronauts, which is really good, but to bring back hope here on Earth. And this is when we start sending brain organoids derived from people with syndromes. And again, I mean, this is a Rett syndrome. That's a child in Rett syndrome. It stopped talking, stopped losing the motor coordinations, and this child will end up in a wheelchair and will stay there for life. So when we send brain organoids from Rett syndrome, we saw something quite unique. We saw an overexpression or something that wasn't there related to a viral activation. Okay, we never add any virus on those tubes. Where is the virus coming from? The virus is coming from inside your genome. Your genome contains a vast majority of sections that are related to old retroviruses that accumulate throughout evolution. And we, in a normal situation, a health situation, we keep them silent. But in a stressful situation, these endogenous retrovirus, how we call, start to become expressed, start to emerge in the cells. And when the cells see that viral particles inside them, what it does, it thinks that it is being attacked by a virus and it triggers an inflammatory pathway. And that's one of the cytokines that is related to inflammation, IL-6, is highly elevated in Rett syndrome compared to controls. So now, if the inflammation that is neurotoxic is coming from endogenous retroviruses, can we use an antiretroviral to treat a neurological condition? And we thought, well, I mean, sounds crazy, but we can actually test that. So we went back to the mouse model for Rett syndrome, which is not a good one because it's so dramatic. The mouse model for Rett syndrome dies in 4 weeks. In 4 weeks, the animal dies. One day before the animal dies, we start treating them with this antiretroviral, and the animal survives. We extend the survival rate of the animal, which would be equivalent of 10 years in humans. And not only that, but the quality of life of these animals start to improve again. So the animals were lethargic. Again, they were about to die, right? They were in the corner, not eating anymore. But after this treatment, they start recovering. And we haven't done like a tremendous follow-up on that yet to see, I mean, how much can we improve? But nonetheless, this is enough for us to start like a clinical trial using one of these anti-HIV common drugs to repurpose for this condition. So that's kind of the pathway that we are going now, trying again to help astronauts, but also bring back the knowledge to help people here on Earth. And when we start presenting this data, many people in patient associations start to ask, what if you can use the same technology, but now looking for late onset conditions such as dementia, Alzheimer's, Parkinson, ALS. And that's a big market, and that's also a big health problem in the world. As we age more, these aging-related conditions are getting more and more common. So it's of interest of many countries to have drugs that can actually block these age-related conditions. And I thought about how to do that. And and to me, the first thing that we needed to do is to have a good chemical library. And that when, for a short description, I was going through an emotional, professional, personal crisis when I first started dealing with the shamans in the Amazon and started getting like exposed to ayahuasca and other medicines from the forest, and I started talking to them and learning what they do. And then I realized that, all right, so perhaps I could use the pharmacology that is already existing in the biodiversity of the Amazon to test, to create new drugs for these type of conditions. And I'm going to show you a video that kind of summarizes the whole process. Basically, the stage where we are, we have this collaboration with the University of Manaus in the middle of the Amazon, where we go and we talk to the original tribes. Most of the collaborators that we have are from the Huni Queens, but we are also talking to the Yanawa and other tribes as well. And we go and ask them, what are the plants that we use when someone starts forgetting things? And they come and show us, oh, these are the plants. And guess what? Nobody knows about those plants. So it's totally unknown. Actually, I remember now when I was in college taking like biology classes, all the professors, they were saying, oh, don't even think about using these molecules because it's so complex, so complicated, and nobody knows exactly how they work. So we were disencouraged to study the biodiversity of the Amazon. But I wanted to go back and to my oranges as a Brazilian and actually go there and try to see if there is something we can do it. And just for the records, we are doing that in collaboration with them. If any of this medicine actually becomes a drug, and royalties will go back to the protection of these tribes and to the protection of the Amazon. Thank you. Thank you. There was a problem, right? Because when you go to these tribes, not everybody knows about these plants. It's really the old shamans who actually have the knowledge. So the first thing that we need to do is to learn from them. And to do that, we are recording all the process, when they choose the leaves, when they choose the plants, how they create all this brew. And we even place like EEGs on their brain so we can record what they're thinking, what they're focusing on while we do that. And then some of these plants are not easy to find, right? They are always spreading to the Amazon. So how are we going to learn that? So we create like a small chip and we place that inside a biodegradable origami robot that we spread out through the Amazon, and they have sensors that can locate the plants that contain neuroactives. And they tell us the location, so we can send members of the tribe to go there. They collect some of the plants, and they replant more. So we preserve the niche, the same location where we found those plants. So we bring to the lab, we purify those molecules. There is a large group of young scientists at UFUN, the Manaus Federal University, who are using different AI tools to predict where they're gonna bind into the receptors of neurons. We bring to San Diego, to the lab in San Diego, we test them, and if they pass the test, they are high candidates to go to the space station so we can test it. And we create a new way to actually test the efficacy of these neuroactives at the International Space Station. Remember, for the organoids, for us to record the electrical activity, they need to be attached to the microelectrodes. Well, in space, there is no gravity for them to fall down in the electrodes. So we have to be creative and rethink the way to do that. So this video, we actually show that. We can just let it run. I'll explain what's going on. So yeah, so the idea is that we are focusing for this specific project on Alzheimer's organoids. And I'm gonna show you some of the contact that we have these original tribes. So these are some of the places that we go close by Manaus. These are some of the tribes that we are working with them. By the way, this is guaraná. It looks like an eye. I like it because it has lots of neuroactive compounds never studied before. We just know the one that keeps you awake, but there's tons of other molecules in there. So these are the plants that contains the DMT. These are the things that they do. So they don't only collect and make a tea. There is lots of things. So this is the EEG cap that we are placing on them so we can measure how they do it. This is the tiny origami robot produced in Japan. We create like a swarm that flies to the Amazon. We tried to parachute them, but they were getting into the trees, so it didn't work. So for a while, they just fly through, and they have this AI recognition that tells exactly when are the plants. So once we collected, purify at UFUN, these are like checking the docking on the neuronal receptors, bring it back to San Diego. We are testing those into the brain organoids before they go to the space station, and making sure that they are working as supposed. And again, I mean, this is the project that will go into a SpaceX rocket, go to the space station, and I'll show you exactly how we prepare to measure that. It's inside a cube like that. Each one of these cubes contains many pieces. The pieces have like a tower that contains microfluidic chamber. Now the electrodes are squishing the organoid so we can measure the electrical activity while they are in space, and we can inject the neuroactive drugs to see how much they become neuroprotected or not. So we can visualize all of that in naked eye, but you can also have three-dimensional microscopy to see even neurons inside of the network, how they are behaving there. So we think that this is a kind of a project that we cannot do it using like a single box, the autonomous box. It requires an astronaut to do it. And we have been in touch with different astronauts, but the problem is that it is almost like asking someone to perform a neurosurgery in someone by just telling them what to do. It's very hard for an astronaut to follow the instructions for Earth for them to do. And because of that, we are teaming up with other nations to create the first cohort of scientist astronauts. Yeah, so we want to make the scientists, the astronauts, to go to the space station or other platforms and actually perform these experiments by themselves. So we don't have to train an astronaut, spend so much time to do that. And applications for that goes for more than like just drug screening, which I show you, but rejuvenation, regeneration, and even understanding health aging. If we can do that, I think we are in really good shape. And I'll finish here with one of the donations that we received from Sanford to UCSD to create the new institute that we have, and this space center is part of this new institute. And I'll end with like a futuristic view of the first scientist, astronaut at the space station growing these brain organoids to treat all these neurological conditions here on Earth. These are my funding agencies and members of my lab, who are so many to mention here. And I would like to invite you, if you want to continue this discussion, my team, are hosting a happy hour that is going to happen on the 10th, and you are all welcome to join. Thank you very much. Thank you. Thank you, guys. We might have time for 1 or 2 questions. If someone has like a burning question, I'm happy to. The microphone is here.
Thank you so much. This was amazing. I have one question, because you work with the indigenous people there, and I was thinking, have you ever considered using traditional Chinese medicine knowledge, which is already 1,000 years old, which maybe also...
You are right, and now, I mean, I feel that this project is becoming so much international. So I just had like, I presented like the data from Saudi Arabia, and they said, we want to do the same thing with the coral reefs. So they are losing the coral reef, is bleaching there. So they are collecting samples to extract the biomaterial from these coral reefs, because I mean, once they're gone, it's gone, right? And they think that this might be like a nice project to do the same thing. So Chinese medicine and other medicines that we modern scientists have been ignoring for so long, and they already know that it works. So it's just a matter of finding the mechanism of action so we can leverage that. Yeah, so it's the combination of ancestral knowledge with the modern science. That's what I want to do. Yeah.
Hello, and thank you so much for the presentation. I was curious, everything you presented or like most of what you presented, has to do with rehabilitation or fixing any issues or addressing a lot of the diseases we see every day. Are you doing any research that has to do with like enhancing cognitive abilities, adding what's possible, thinking about the future? I'm curious about that.
Yeah, so we do. This is a top secret, by the way, but but we do. Yeah, so part of the evolutional aspect of the lab is to create brains that never exist. Even the format of the brain we can change, and that might create brains that compute better. We can even create, I never mentioned here, but what we call chimeras, where we mix brains from other species to achieve the power that that species have. For example, if you Google chimpanzees and memory test, you're going to see how a chimpanzee can memory in a very short period of time, tons of numbers and sequences that you cannot even imagine, but they do it. How they do it? So can I incorporate that into human brain? Can I plug that into human brain? Another project that we have, it is to recreate the magnetic sensors in our genomes. At one point in evolution, similar to whales and other animals, we had that, but we lost it. It's all mutated in our genome. So I'm reconstructing that to create a brain that can sense magnetic fields. Yeah, so we are on that.
I have 2 questions. The first one is, have you tried conglomerating the organoids to get to some point where there's consciousness, and then using that as a vehicle for experimentation? And then the second one is, have you used AI to accelerate or not have to go into outer space, just do it through a use of AI?
Yeah, so I will start with the second one. Yes, this is very intriguing. We are trying to apply all the AI tools to see if there are pathways that we can manipulate to accelerate aging. So far, everything that we try has a detrimental consequence. So you can accelerate, for example, synaptogenesis, but the neurons will die earlier. So we haven't found the balance yet, and it might be that's not possible, because evolution probably tried that for millions of years and also couldn't do it. But we keep trying. And the second question about consciousness. So there is a huge debate. It might be that the organoids that we have, even though they're not connecting with a network, they might already be conscious. So in 2019, when we published that data, I invited several people from the philosophy, the ethical fields, philosophers of the mind, to discuss the data and to see if we need any kind of a supervision from the ethical side. And the discussion ended up being about consciousness. Are those structures conscious or not? And there are some people, for example, Pat Churchland from UCSD, she doesn't believe that those structures have any signs of consciousness. On the other hand, you have someone like Christopher Koch from the Allen Institute who believes that they are already conscious. It's just a matter of which level of consciousness they are. So we did like one experiment with Stuart Hemmerhoff, who also into conscious. He proposed an experiment which is to anesthetize one of these organoids and see if the brain waves goes away, because that's exactly what happens in the human brain. So if we add the anesthetic and the brain waves continue, they are not conscious. But the result was the opposite. When we add the anesthetic, the brain waves silence. And then we remove the anesthetics, they're back. Which is not like a proof of consciousness, but is an evidence that they might have some level of consciousness already. So if we start plugging them, they might reach even higher levels.
Thank you.
This one.
Hey, real quick. If I understood the lecture, you were saying that in these organoids, even without the presence of structured stimuli, synapses are still formed.
Yeah.
Can you just explain, I guess, what kinds of connections are being formed and what do they look like?
Yeah, so it is true. I mean, even with normal maturation, they are finding each other and forming synapses. So these are a mix of chemical synapses as well as electrical synapses. I mean, we can actually see them. Exactly how they are forming that seems like a mystery to me as well. It might be that they are creating their own input, we just don't know. So that might be like in the forms of neurotransmitters that they are releasing, things like that, other factors that we don't know. That's why I like the model. It's actually opening new questions about how the human brain is formed that we never had the opportunity to look. All right, I think my time is up, but thank you so much.